we're ready, hopefully. So yeah, thank you so much for the kind introduction, and thanks to the organizers for letting me present today alongside so many friends. It's really exciting to present to so many friends and also to meet new friends, hopefully. So yeah, my name's Allegra. I'm coming to you all today from University of Denver, where I'm an assistant professor. And so I'll be telling you a little bit about what we do in my lab, and that is as Joey said, find or try to find new undiscovered metal binding molecules using methods like electrospray metabolomics and thinking about going beyond this method. And so in my talk today, I'll be starting by describing this method, which we developed when I was a postdoc in Peter Dorstein's lab. I'll describe a story from when I was a postdoc, and then I'll next move on to describing a story from my own lab. But all right. I like starting by showing this image because this is one of my favorite places in the greater Denver metro area. But also I like this picture because I want to remind you all that metals are always all around us, right? So there are metals in rocks. There are also metals in plants, right? And of course there are metals within us, right? And so I want to remind you also that metals are essential for life. They're not only within us, they're also essential, right? Because metals carry out functions like respiration and electron transfer, oxygen transport, along with DNA replication and repair, and photosynthesis, along with nitrogen fixation. So metals aren't only essential for us to live, they're also essential for microbes to survive. And so the stories I'll be telling you today are about microorganisms that live in our gut and microorganisms that live on leaves in the phytosphere microbiome. But before we dive into this, I'd just like to remind you how microbes actually acquire metals. And so microbes have the really cool ability to biosynthesize small molecules, natural products, right, that combine metals with exquisite selectivity and specificity, right? And so in the case of iron, these are called siderophores. And in the case of other metals like copper, zinc, or even rare earth elements and lanthanides, which I'll be describing soon, these are called metallophores. So Vic, in his talk, sort of foreshadowed a bit of this today. By describing some potential metal binding molecules, but essentially microbes can secrete these molecules into metal deficient environments where they can bind metals and deliver them back to the microbe. So as Joey mentioned, I have always been really fascinated by metal chemistry, but my lab is really interested in metal binding molecules, first because we think the chemistry is cool and exciting, but also because these molecules play really important roles across biology. So they can mediate microbial interactions, they can mediate interactions between host and microbes, and they can also do this in plant systems, in the ocean ecosystem, and we think that perhaps we can even use these molecules for energy, recycling, and bioremediation applications. And so the first story I'll tell goes back to when I was a postdoc in Peter's lab. So I was really hoping to answer this question. Essentially, how do metal binding molecules shape interactions within host-associated microbiomes? Now, it quickly became apparent this was a very ambitious question to answer. So now I hope to answer this in my whole career. But at the time, we very quickly realized that high throughput methods for finding metal interactions with small molecules were limited. And so with my colleague Daniel, who is sort of another advisor when I was in the Dorstein lab, we hypothesized this was due to a few different reasons. First, we thought that low pH and high concentrations of organic solvent that we use for chromatography and sample extraction can destroy metal complexes. And also, when we do our metabolomics preparations, we use low metal concentrations, right? And so this disfavors equilibrium away from metal complexation. And then finally, because these molecules bind metals so well, they're often produced at lower quantities. So our signal intensities can sometimes be close to detection limits. So Daniel and I hypothesized that we could make some pretty simple modifications to our standard LCMS-MS workflow to allow us to capture and find these metal binding molecules. we first thought we could take advantage of altered MS conditions using a more native ESI approach, and we could perhaps use novel computational strategies to develop a method that we called native metabolomics. Okay. And so let me show you how this works. So here you can see sort of our schematic. We run our chromatography at low pH, but then after chromatography, we can add a buffer to raise our pH to some physiologically relevant level. Okay, and then at this point, we can infuse in our metal salt of interest. Now our metal salt can bind to molecules that are metal binding. And so just real quick, I want to mention the following thing. Because we've designed this so that we're infusing metal salt after chromatography, we expect to see both the proton bound and the iron bound version of the molecule occurring at the same retention time. So to test if this could work, we took 5 standards shown here, and we just tried out this experimental workflow on these standards. Now you can see in the case of ferrochrome, when we do not use this workflow, when we use standard LCMS conditions, we don't see the iron adduct, right? We only see the proton and the sodium adducts. But then using our new workflow, we see both the proton bound and the iron bound adducts. Okay, so this was not super surprising, but it was very exciting because it suggested that we could actually start applying our method to more complex samples. And so how are we going to do that? Well, we thought we could take advantage of feature-based molecular networking and ion identity networking. So luckily, I don't need to describe feature-based networking because Vanessa already did, but I will briefly describe ion identity molecular networking. So the way this works is we can do feature finding, which again, Vanessa described, and then from there, we can perform retention time and peak shape correlations, and then a subsequent filtering step. So if we infuse in iron, we can filter to look for all of the correlated features that have the iron-specific mass delta. And once we find all these features, we can add this MS1 ion identity edge, perhaps adding it as an orange dashed line to indicate the presence of an iron binding event. And so you can see on those 5 standards, this worked very well. And now you're seeing in this feature-based molecular network that each of the siderophores are like jumping out at you out of this molecular network. So now I'd like to turn to tell a story that actually started long before my postdoc with this organism, E. coli Nissl. Now, this is a probiotic organism that was actually isolated from the gut of a soldier in World War I. But we became really interested in this because we thought that perhaps one of the mechanisms through which this acts as a probiotic is through outcompeting other organisms for metals. And one of the reasons we thought this was because of experiments that were done by our collaborators, Manuela Raffaello and her postdoc, Huey Z. Okay, and so here what you can see is that E. coli Nissl is actually more resistant than Salmonella to zinc limitation. You can see this here, where increasing levels of the zinc sequestration protein calprotectin result in decreased levels of zinc. And you can see at the highest level of calprotectin, or the lowest level of zinc, E. coli Nissl outcompetes Salmonella. Okay, so you might ask, well, how does E. coli Nissl actually acquire zinc from its environment? So it has 2 zinc transporters. Okay, but the interesting thing is that when both of these are mutated and are unable to let zinc into the cell, you actually still see that E. coli Nissl outcompetes Salmonella. So then this suggests there might be another zinc acquisition mechanism. And so what we did at this point is we looked through the genome and we found there's a putative metallophore biosynthetic gene cluster. Okay, so now we hypothesize, well, maybe this is actually producing a putative metallophore that can be taken up by some other protein. So in order to test this, Manuela and Huey made the mutant of IRP2, which is the synthase that makes this putative metallophore. And we ran wild type versus mutant samples to generate this large molecular network. And so now what we're looking for are nodes that are only found in the wild type but not found in both the knockout and the wild type. Okay, and so those are pink notes. So I want to draw your attention to this large molecular family. And so you can see they're mostly pink nodes. So we were quite eager to look at these larger nodes, right? Because here we've scaled our node size by feature abundance. And when we looked at these nodes, we found that they are this molecule, which is Yersinia bactin. We were really intrigued because Yersinia bactin is a known siderophore. So we wondered, could this actually be a promiscuous metallophore that also binds zinc as well? And so to test that, we had our new method developed. And so what we did is we ran a post-LC infusion of zinc after pH neutralization. And we were quite excited to get a molecular network. And here I've zoomed in on the molecular family containing Yersinia bactin. And so I want to draw your attention to these blue dashed edges, which now correspond to those nodes that differ by a zinc-dependent mass delta. And so what we found is that Yersinia bactin here exhibits both the apo and the zinc-bound complex. And additionally, we found various other derivatives that also seem to be binding zinc. We were able to confirm this by NMR, but in the interest of time, I won't show this results. And I will simply say that we found that this molecule Yersinia bactin also facilitates E. coli nissl colonization of the inflamed gut. So this was work done by our collaborators in the Raffatelli lab. And what you can see here is a higher ratio means that wild type exhibits a significant growth advantage over the mutant. And so we're seeing that Yersinia bactin is important in vivo. Okay, and so now I've kind of raced through a story about a zinc binding molecule, but I really wanted to get to this part of my talk where I can tell you that in addition to looking at many more siderophores, we're also really interested in extending our search for metal binding molecules that bind metals across the periodic table. And one of the stories I'm most excited about is actually a story that emerged through this collaboration with Ceci and Alexa, looking at lanthanides in biology. So maybe some of you aren't aware that lanthanides are even involved in biology, because no one was aware of this until about a decade ago, when it was discovered that methylobrum extorquins actually uses lanthanides in addition to calcium for its methanol dehydrogenase enzyme. Okay, so this organism can metabolize methanol. So there's been significant work in the past decade to understand lanthanide homeostasis in this organism, but it still remained unknown how the organism could actually acquire lanthanides from its external environment. And so this began with work done in the Gomez-Martinez lab, the Martinez-Gomez lab. And so here you can see the results of transcriptomics, where these organisms were grown in the presence of a soluble lanthanide source versus an insoluble lanthanide source. So in the case of the insoluble lanthanide, this is sort of like a lanthanide limitation. And what they observed was that we see the expression of all of these genes here, okay? Now these are all in a gene cluster that looks very similar to a metallophore-like gene cluster, okay? And so we hypothesized that maybe they were responsible for producing a lanthanide binding metallophore. And so what our collaborators did is they made the mutant where they knocked out, actually they knocked out the whole gene cluster. They also made various other mutants, but then we were able to test wild type versus overexpression versus our knockout. And so you can see the results of our metabolomics experiment here. And so now what we were looking for was for a molecular family that was only present in the overexpression, or the blue, and the wild type, or the orange and the red, but not in the green. And we're very excited to see just one molecular family. Now, when we looked more closely at that molecular family, we found that one feature seemed to be really significant and highly differentially abundant in the wild type versus the knockout. And when we looked more closely at the MS2 for that, we were actually able to annotate that as this new molecule, which looks similar to a known siderophore, protopetrobactin. And so we were quite excited because this allowed us to identify the first biological small molecule lanthanide chelator. And so now I just wanted to highlight my student Marquise, who's here, because he's following up on the biosynthesis of this molecule. And also we're starting to look more closely at various other organisms in this family to try to see what is the chemical diversity of this class of molecules. And so with that, I'll summarize. We've developed new experimental and computational approaches for discovering metallophores from biological systems, and we've started applying these tools to find new and what I think are exciting things. And so with that, I really want to thank both my lab and also Peter's lab and all of my collaborators, many of whom are actually here, because this work would not have been possible without all of these collaborators and without all of these people too. So thank you guys for listening. And yeah, I'm happy to take.
Sure. Yeah, great talk.
Thanks.
I was curious if you have a strategy for like how many metals can you co-inject at once, I guess? If you have a strategy for identifying like specific targets, or do you have a metal in mind and then you search, or do you have lots of metals and you're going to see what sticks?
Yeah, no, that's a really good question. So usually we do sort of have a metal in mind, but often we are interested to see like how promiscuous is the molecule, right? Because with ursinobactin, for example, it was discovered as a siderophore long, long ago, right? But then reports came out that it actually binds copper, and that was biologically relevant. And then we found it binds zinc, and that is also biologically relevant, right? So, so we usually test one metal, but then we move on to doing a panel. So in principle, we can inject maybe as many as we'd want. I think we've only done 5, but the thing is, if we inject so many metals that our salt concentration gets really high, we do see ion suppression sometimes. So maybe we wouldn't want to inject like 30 at a time, but I think, yeah, we've tried 5. You can probably go higher, but we do often try to prioritize, yeah, based on also some growth experiments. Yeah.
Can you use the ratio of M plus H to M plus metal to get like a faux equilibrium binding coefficient for the system?
Yes. Great question. And that is something that a lot of people ask, and I wish that were the case, but we cannot. I think because, well, we can't do that, but I'm thinking maybe we can do some look at MS2 and fragmentation and try to figure it out that way, perhaps. We haven't started doing this, but it's something I'm excited to start. Because yes, everyone wants to know KDs and, or at least relative sort of dissociation, constant strength of binding, and I would like to also, but we can't do it that way. Yeah.
When you're doing the correlations of MSMS, how well does MSMS do at pulling together the different adduct types? I can imagine those would be very different.
Yes, so MSMS does not usually do very well at pulling the adduct types together. That's why we're looking rather at the MS1, where we're looking at retention time and peak shape correlations and mass deltas. Because yeah, you're right that the MS2s of like iron versus proton are often quite different. Sometimes they're not, but most of the time they're very different.
And then a follow-up to that, have you started mining why they're, or what's changing? What's like, do you think that you can figure out where those metals are actually binding by doing comparative analysis of the MSMS?
Yes. Well, this is something I'm quite interested in doing. I'm not sure if it's possible, but I have wanted to do this for a while now. I just haven't got to it yet. So, but yeah, that is something I'm very interested in doing. Yeah. Yeah, Nina.
When you infuse the metal to the instrument, it is causing any trouble to the instrument?
we clean our instrument maybe more routinely than when I wasn't doing it all the time. So maybe not exactly, but yeah, we do clean our instrument a lot because we do see like like we do see kind of like a ring of very dark stuff on the yeah.
Maybe
Daniel.
to add on the spectrum of infusing weird things in your mass spec is not as bad as directly infusing seawater.
But we do actually see some like iron, I forget what exactly, I think iron something clusters. Like there are, Marquise might be able to answer specifically what type of cluster we see, but we see like key contaminants that are iron, sort of iron contaminants if we don't wash well enough.
Sorry, how do you control for oxidation states of some of those big transition metals?
Yeah. So we're typically we're typically infusing like iron 3 instead of iron 2, for example, but we do see some reduction like in the source. So sometimes we look for both actually, iron 3 and iron 2. I guess we don't control for oxidation state, we just look for both. Because we're not looking at like iron 2 binders, for example.
Maybe a similar question is, do you ever see dimers of the siderophores and like spurs
We
forming?
do, yes, we do. Yeah, so that's something also that's really nice with MZ-Mine is we can look for like a dimer, or we can look for like a 2 to 3 or like a 3 to 2 or 2 to 3 complex, right? Which can be the biological way of chelation, right? So yeah, so we do see that. Yeah. And we see it all.
Great, let's thank Allegra again.